Good afternoon, everybody. Thank you so much for the invitation to be here this afternoon to present to you. My talk is slightly different to everyone's, but in some way actually incorporates a lot of what many of our speakers have said throughout the last 2 days. I worked at the ARC since up until February last year for about 5 years, managing the invasive species or the Biocontrol unit investigating management of fynbos weeds. And I'm currently based in Australia, but I remain a research fellow at the University of Cape Town. So what I'll talk about today is a bit of research that's been done in South Africa to actually manage risks to biodiversity using insects. And obviously this is looking at the biological control of invasive plant species. Okay, now we have to get it to go. Oh, what is happening? Did it move?
Nothing, still on the first slide.
Okay, there we go.
There we go.
Okay, fabulous. All right, so we've had a bit of a great introduction from Professor Wingfield yesterday in terms of invasive species and pathways. From a few speakers have actually spoken about this. And in brief, invasive species come from other countries via various means, and often this is through trade routes or the importation of wood. This was also spoken about by Wilhelm in terms of the polyphagous shottle borer. And biocontrol is a process of managing these invasive species, whether it's an insect or an animal or a plant, but for the purposes of this talk, I will be talking about the biocontrol of invasive plant species. So biocontrol uses natural enemies to control invasive weed species. And often the natural enemy, well, sorry, in most instances, the natural enemy originates from the same population of where the weed comes from. So in our instance, a lot of our invasive species that we dealt with down in the Western Cape originate from Australia, and often the agents that we use to manage them would also originate from Australia. So the main purpose of biological control, which is quite important to remember, is that it's not to eradicate the weed species of concern, but it's rather to try and reduce the population or to manage the population of the weed. A few really good things about biological control is that it's considered self-perpetuating. It's a long-term and a very sustainable, environmentally friendly solution to invasive species management. And it's relatively inexpensive when compared to the traditional forms of managing invasive species, such as manual clearing or spraying of herbicides, pesticides, or insecticides. So for the rest of the talk, I will be speaking a little bit about what the aim of some of the biocontrol programs within South Africa and globally actually are, and give you a few case studies in terms of what has been achieved and how this is assessed across different landscapes. So in general, there are 2 biocontrol strategies that are implemented in order to manage invasive weed species, and these are often reliant on whether or not the species that we are looking to control is considered beneficial or not. I know that forestry species are considered beneficial, so in this instance we would not use the density reduction strategy, which is the first one I've listed on the slide here. This sort of strategy is used when the weed has got no beneficial properties. And often the agent, when I speak about agents, by the way, I'm referring to either insects, pathogens, or mites, which have all been utilized for biocontrol globally and within South Africa. So the agents that we use in a density reduction strategy would often attack the photosynthetic and structural tissues of a plant. And obviously, this would negatively impact how something looks. So then obviously that plant would not be aesthetically pleasing if it was one that was utilized in a wood industry, for example. The agents utilized in this sort of strategy would actually have the ability to kill the plants and therefore reduce the overall density of that invasive plant species. The other strategy that is utilized more often in a case where species are considered beneficial would be one of containment. And this is often utilized in good examples of these, which I think some of you may be familiar with, would be the management of prosopis in the Northern Cape in particular, as well as wattles throughout the South African region. And the containment strategy often makes use of agents which attack only the reproductive parts of the plants. So the main purpose of some of these strategies would be to reduce the overall reproductive output of the species that are being targeted. So the weed will not be killed by the agent's actions, but rather the future spread will be curtailed. Great, that looks better now. All right, so in South Africa, there's a procedure which is utilized by a variety of different stakeholders, particularly researchers within the field of biological control, which assigns various levels of control to biological control programs. So this means that we assess whether or not a species, a weed species, for example, would be considered under complete control, substantial control, or negligible control. And often this is a very subjective, you know, as the nature of humans assigning a category to something, it is subjective and it is fluid. But it is informed by science and practice. And as research evolves and as we utilize or investigate additional agents to manage specific weed species, the level of control assigned to a particular weed species may change. So those are just some of the publications that have dealt with how we assign levels of control for some of the plant species that we work on within South Africa. This is just a photograph to show you what a typical Western Cape mountain slope might look like. This comes from, I think it's from the Stellenbosch region, but it's somebody else's photograph. I think it was John Hoffman's photograph. And it's just to show you some of the most critical invaders in the fynbos biome that we have to deal with. And in the foreground, we've got acacia species, and you can see that many of these are young plants. This was taken after a fire. What often happens in situations like this is that acacias and hackeias are extremely fire adapted. So the minute a fire comes through, all the seed bank, the seed bank germinates, and you end up with a potentially much huger problem than what you had to begin with. You can see in the back there's some poplars there, and often these species will grow in watercourses along with the eucalypts on the left-hand side, which is also growing along the riverbank. Hackeias and pines generally would grow on the mountain slopes and would outcompete some of our indigenous fynbos species, which obviously all of these species impact biodiversity of native flora quite significantly. So now I'm going to move on to a few of these case studies utilizing this sort of classification, I guess you could call it, in terms of how successful or not successful control programs against some weeds have been in South Africa. I thought I'd start off by using one which many people might be familiar with, and that would be the case of Sesbania control. The Sesbania tree is now considered to be under complete control as a result of the actions of 3 biological control agents that were introduced for its control many decades back. There are 3 weevil species that are utilized for managing Sesbania, and the bottom right-hand one, that red, stripy, black and yellow one, is a very beautiful weevil species, and it's a stem borer and has actually does have the potential to actually kill trees. So as a result of the combined impact of these 3 species on Sesbania stands, we effectively have now got complete control of Sesbania in Southern Africa, in the Western Cape and the Eastern Cape. And this is a slide from John Hoffman, which shows what a Sesbania infestation looked before my time, way before my time in 1985. And then once biocontrol had been released and all 3 agents, which is quite critical for this system, had been utilized and worked together, what it looked like about 10 years later, where complete stands of the trees had actually been decimated as a result of feeding action from these weevils. So another group, I guess, it's not consistently the case across the cacti group, but many cactus species have experienced levels of substantial control in terms of the biocontrol programs within South Africa and Australia. And an interesting thing is that one of the first biocontrol programs, or the first biocontrol program utilized internationally and within South Africa, was one against Opuntia species. And it was so effective that it decimated, or should I say, cleared vast stands of prickly pear at the time in Australia, which I think set up the biocontrol fraternity for sort of a catch-up and constant questioning as to why does the system not act in the same way as what the system did against prickly pear, for example, because it was so successful that I think everybody expects that that is what the situation would be like for every weed that we try to manage going forward. However, that's obviously not the case. So in South Africa, Opuntia stricta in the Kruger National Park caused significant issues in terms of biodiversity impacts. And the images on the right-hand side show how dense an infestation there actually was at some point before the release of a cactoblastis moth and a cochineal species, which then worked very well together to be able to clear the site of Opuntia stricta within 20 years of their initial release. So lots of prickly pear has actually, or should I say Opuntia species have actually impacted several agroecosystems as well. And a really, one of the most recent ones that I know or that I know a bit more about is the boxing glove cactus, which was, I think, first identified early or late 2012. And a biocontrol program was initiated against boxing glove cactus, and it's to this day considered now to be under complete control. That's how effective that program was in such a short space of time. So again, oh, no, it's not moving. There we go, sorry. So this is again an old slide from before the implementation of biocontrol in 1939. The top slide shows really high, thick, dense stands of prickly pear in the Graaff-Reinet region. Once biocontrol had been implemented, less than 20 years later, you can see that this entire site has basically been cleared of invasive prickly pear. So I don't want this to be an indication, or I don't want this to be taken as biocontrol is extremely effective against cactus species, because it's not. There are several cactus species for which biocontrol does not work really well. And there are several factors that impact the success or failure of biocontrol programs, which I'll get into briefly at the end of my presentation. But yeah, I just thought it was a good idea, good schematics of before and after, which unfortunately is one of the things that has been lacking in biocontrol research to date, is an indication of what a system may have been like before the initiation of a biocontrol program against a targeted weed. Another example of what we would call substantial control, which now requires an integrated management strategy to manage the weed. So complete control is a strategy that does not require any other input from management. Biocontrol is doing a sufficient job in order to curtail the spread or the density of the target weed. Substantial control, on the other hand, requires additional inputs from other means, whether this is manual clearing or spraying. So this example, or these examples, are of Acacia longifolia, longleaf wattle, and of Acacia pycnantha, the golden wattle. What's quite interesting here is that we have a tiny little wasp called Trichogasta species on each of these acacia species, as well as a seed-feeding weevil. So you've got two agents that generally don't negatively impact each other, but they work together to try and reduce the reproductive output of the plant as much as possible. So These little wasps lay their eggs inside the vegetative or flower buds of both these acacia species, and instead of these acacias then forming dense seed pods, we instead have these very characteristic galls that are formed either on the stems or in place of where the seed pods would develop. If you bisect one of these galls, you're able to see that there's 3 distinct gall chambers inside each, and each of these gall chambers houses a wasp larva. So the tree then effectively becomes a breeding ground for the wasp rather than a breeding ground for itself. Another acacia species that we consider to be under substantial control is black wattle, Acacia mearnsii. This is a species that definitely still requires integrated control. Again, we have got the seed-feeding weevil melanteris, which you can see on the bottom left of the slide there. This weevil comes in when the pods have actually managed to develop or escape the impact of the other biocontrol agent, and it then feeds and oviposits inside and on the pod to reduce the number of seeds that are actually viable within that pod. Perhaps one of the most prolific, I guess, and popular agents within the fraternity, as well as with other stakeholders such as farmers, would be the gall-forming midge dacinera, which you can see on the top left there. It's a tiny little fly, and it acts in much the same way as what the wasp does on Acacia longifolia and golden wattle, in that it lays its eggs in the flowers of black wattle, and instead of those flowers forming really dense seed pods, as you can see in the middle of that top row of images, it forms these really dense galls or gall clusters, which is the image directly to the right of the midge. And each of these gall clusters actually houses several dozen midge larvae. The midge is really widely distributed in some regions, and particularly in the winter rainfall region, and it has been shown to effectively reduce the seed set of black wattle by up to 99%. So another system where biocontrol is really popular is in the management of invasive water weeds. And one of the species which has been a really good flagship species for biocontrol is parrot's feather, Myriophyllum acutatum. There's only one agent that is utilized for the biocontrol of this water weed in South Africa, and that is a beetle, Lysathia. It's a highly effective species, and it's able to actually clear a water body within just a few years after its release, basically. It's a really good example of how a biocontrol program works, in that it's able to decimate a population of parrot's feather. It then dies down, I guess would be the term, to a lower level, the plant, when the plant is died out, and then the plant obviously then would be able to reseed or resprout from little fragments that may have escaped its impact during season 1. And then the plant flourishes again, and there's a slight lag phase behind it where the insect has to catch up. But for all intents and purposes, it's a species that is under complete control. I included this one in particular because, along with some of the other waterweed species, most of them actually, and several of the cacti species, mass rearing is utilized in order to ensure that we get hundreds of thousands or thousands of insects to be able to release at a time across different locations within that or targeted locations, should I say. So we either make use of mass rearing strategies in specialized facilities to be able to release insects, or we have to collect them in the wild from established populations, either within South Africa or at their source or native population overseas, and then import them into South Africa. Obviously, this has additional constraints in that they have to be reared through in quarantine, and we need to ensure that no other insects or pathogens escape with them when we actually do eventually release them into the wild. So one of the species I focused most of my time on during my 5 years at the ARC was Hackia sericea, or silky hackia, which is a significant invader of the fynbos biome in the Western Cape. It's one of the longest running biological control programs in the country, but it's also the first program globally to actually utilize a seed feeding agent to reduce the spread of an invasive plant. To date, there's been 5 insect species that have been released on Hakea, and we also utilize an indigenous fungus that has been developed into a mycoherbicide by colleagues at the Plant Pathology Unit at the Agricultural Research Council Plant Pathology Unit. So this is an indication of some of the insect species that are most effective on Hakea. On the top row, we see Erythena conspita, which is the seed-feeding weevil, and it feeds on young fruits of Hakea, which then end up looking like the far right-hand image, and we call that a aborted fruit. The middle image is a stem-boring beetle. Sorry, it's a longhorn stem-boring beetle. It lays its eggs on the outside of a Hakea stump. The eggs hatch and the larvae burrow into the stump, and you can see that very characteristic sawdust-like exuvia, which is evident of the presence of the stem borer. So it's a species which unfortunately spends a large amount of time inside the Hakea plant, and in areas where the ecosystem is really prone to fires, it has significant impacts on the survivability and long-term sustainability of this insect for the biocontrol program against Hakea or of Hakea cerevisiae, should I say. The top two,
Candice?
Yeah, no problem. The bottom 2 images are images of also one of the longest running agents on Hakea. So that, along with the seed feeding weevil above, have been in use, I guess, for over 50 years already. The seed feeding moth impacts or it attacks the mature fruits of Hakea, and combined with the seed feeding weevil, they actually have the ability to reduce seed set of or seed production of Hakea trees up to about 70 to 80%, depending on site. The last agent, which has been most recently introduced, is the flower bud feeding weevil, which you can see on the bottom 2 right images. And this is the subject of multiple investigations by one of my PhD students, Kelby English. What's quite important to know here for that species, but also for many others, is that like the black wattle midge, which establishes and does really well in winter rainfall areas, Dicomata rufa does not establish beyond the Southern Cape region. So it does not want to establish in the Winelands region, and this obviously poses questions as to why this could be the case. So there are obviously many species where biocontrol has been successful, but there are also others where biocontrol is having very little impact. And in South Africa, as well as I think in some places globally, some of these species are lantana, Acacia delbata, silver wattle, prosopis, as well as the giant Spanish reed, Arunda donax. And the impacts of agents, as well as whether or not they establish, I guess, from the outset, is influenced by a variety of factors, and this includes a host-agent mismatch in some instances, different ecotypes of species. So a lot of the cactus species, you might have cacti that look identical, but they originate from 2 slightly different localities, and they would therefore potentially have 2 different cochineal species that we would then need to collect and do a whole bunch of tests on to be able to utilize them for biocontrol program going forward, because often one won't work on another, for example. Despite these constraints, biocontrol does still remain the most economically viable and environmentally friendly solution to invasive weed management, and it does allow the utilization of integrated means to manage invasive species. So this is by no means an exhaustive example of all the partners that are involved in biocontrol research, but it's a little bit of an indication of some of the collaborators, or should I say colleagues, that I have had dealings with. And it's a big school of researchers that span either universities or research organizations globally. And South Africa is really one of the top 5 countries in the world implementing biocontrol research against invasive plant species. So thank you all for your time, and thank you so much to John Hoffman and Fiona Impson, as well as UCT, ARC, DEA, who's no longer DEA, I don't know what they're called now, sorry, and obviously the Drakensberg Trust for funding. And thank you all for listening.